Welcome. We're going to be dealing with standard 9.4 for IB Biology HL. We're dealing with the effects of interrupted days and nights and plant flowering. A student from 2020 graduating class, Anmal, went and captured this from a Flash program before Flash died. So let's just go ahead and go through it. For flowering plants, to cross-pollinate, they must produce flowers at the same time of year. How do plants that may be different in size or age synchronize their flowering? In the 1930s, James Bonner and Carl Hammer discovered that the key flowering trigger in some plants is the length of night, which explains why a species will flower during a particular season year after year. For historical reasons, plants are described as short day or long day plants rather than the more appropriate long night and short night plants. Day-neutral plants in which the flowering trigger does not depend on a specific length of darkness, or perhaps more common than short or long day plants. In this animation, we will examine flowering experiments performed on cocklebur, a short day plant. So here we have the animation. We have the short day plant with the scientific name that begins to flower in the late summer and fall when day length no longer exceeds 15 hours. In an experiment by Carl Hammer and James Bonner, the cocklebur plants were grown continuously on 16-hour light, 8-hour dark cycles. Under these conditions, the plants remained in a vegetative, non-flowering state indefinitely. Here we see this illustration of 16 hours of sunlight, 8 hours of night, and we see that we have no flowering occur. Now, if we reduce it by 1 hour to 15 hours of daylight and 9 hours of darkness, we then see flowering occur. So here's our first question. Do these results allow you to distinguish which factor is important in flowering? The length of darkness or light? A, these results suggest the length of the light period is a critical factor. B, these results suggest that the length of dark period is the critical factor. C, from these results, it is impossible to determine which period is critical to flowering. Take a moment. Can you figure out what the answer is? Let's try C. Correct. C. From these experiments, it is not possible to determine the critical factor in flowering. Both factors are varied together in this experiment. Now we go on in the experiment in photoperiods that were not on a 24-hour cycle, but extended to a 48-hour cycle. In the new treatment, plants received light during the first 16 hours, followed by 32 hours of dark period. Plants flowered under these conditions. So again, 16 hours light, 8 hours dark, 16 hours light, 8 hours dark, over 48 hours, no flowering, and it's repeated. Okay, 15, 9, 15, 9, flowering, 16, 32, flowering. Okay, now let's look at what happens when other plants received a 12-hour cycle with 4 hours of light followed by 8 hours of darkness. The plants did not flower under these conditions. 4 hours of light, 8 hours of dark, 4 hours of light, 8 hours of dark, 4 hours of light, 8 hours of dark, etc., etc. What can be concluded from these results? Is it A, a light period in 15 hours is or less is sufficient to trigger flowering? B, a dark period of 9 hours or more is sufficient to trigger flowering? Or C, a dark period of 9 hours or less is sufficient to trigger flowering? Take a moment, think about which is the best answer. Let's try A. That is incorrect. Notice that the light period of 4 hours was insufficient to trigger flowering. Try again. Let's try C. Incorrect. In all cases in which the period of darkness was less than 9 hours, the plant did not flower. Try again. Oh, well, I guess that leaves us with B. Correct. For these experiments, it appears that 9 hours or more of darkness after the light period triggers flowering. So it didn't matter whether it was 9 hours or 32 hours. but it had to be 9 hours or more of continuous darkness. The experiment followed to pinpoint the length of darkness as important in flowering in the cocklebur, with 9 hours as the critical length. To test whether a continuous 9 hours is important, they interrupted the dark period with 1 minute of illumination. The treatment resulted in no flowering. So here we see 1 minute of light turned on. So the investigators determined that short day and long day plants have a timing mechanism that measures the length of a continuous dark period. With this in mind, predict whether each of these conditions result in flowering. Click yes for flowering and no for no flowering at the appropriate position in the table above. So a short day plant, should that flower or not flower with this long day, short night? Long day plants should flower. Here we have short conditions. We have day interrupted with a little night, and then we have a short night. So should we get flowering? No. Long day, should we get flowering? Yes. Now we've got a long night, short day. Should we get flowering in the short day plants? Yes. Should we get flowering in the long day plants? No. Interrupt the night, should we get flowering? No. Should we get flowering in long day plants? Let's try yes. We are correct. So you've correctly anticipated the consequences of each light condition for short day and long day plants. Both types of plants measure the length of the dark period. Notice that the long day plants interrupted a long night with a short period of illumination, essentially shortening the night, triggering flowering, just as a short night does. So when we come down to it, we see that we have these phytochromes for red and phytochrome far red, and they go back and forth as far as the molecules are concerned. The nature of the timing mechanism has been partially revealed and involves a pigment called phytochrome. Phytochrome exists in two forms, in phytochrome red and in phytochrome far red, which refer to the red light absorbing and the far red light absorbing forms of light. So remember, red light you find in sunlight and also in a lot of artificial lights. Far red is beyond the human visual spectrum, but still measurable. So if we take a look here, we see that when you have red, converts the red light will convert the phytochrome red, which is inactive, into phytochrome far red, which is biologically active. In turn, far red converts phytochrome far red back into phytochrome red. Also, phytochrome far red also steadily reverts back into the phytochrome red form at night when there's no light. Some of the phytochrome far red molecules also decay. So again, you turn on the light, you get some red light hitting the phytochrome red, boom, it jumps over to being phytochrome far red. Phytochrome far red will, during the nighttime, revert back to being in the phytochrome red conformation. And also we get a little bit of breakdown of it. Now, if you do have access to far red light, you can artificially expose phytochrome far red to a substantial amount of phytochrome far red. Sunlight contains more red than far red light. As night begins, phytochrome is almost all in the phytochrome far red form. Over a period of several hours in the dark, these molecules begin to convert back to phytochrome red because they're not getting exposed to phytochrome, or they're not getting exposed to sunlight. So over a period of several hours of dark, these molecules begin to convert back to phytochrome red or to break down. Depending on the length of darkness, only some of the phytochrome red remains as dawn breaks. If a flash of red light interrupts a long night, the phytochrome molecules reset themselves, converting back to phytochrome red. And so here we're going to take a look at night interruptions have opposite effects in short day and long day plants. And in long day plants, it promotes flowering. Determine how this the series of light flashes would affect the flowering in short day and long day plants. Click yes for flowering and no for no flowering. Okay, so we know here's the night is long enough, so we should get flowering. Here, the night is long, the day is short, so long day plants with we should have no flowering. Here we see that we interrupt the night with red light, which is going to convert all that phytochrome far red back into phytochrome red. So we have essentially turned on the sun. And so for short day plants, we've interrupted the night, so we should get no flowering. For long day plants, we should get flowering. Now, if we interrupt it with far red, we're reinforcing the darkness. We're reinforcing the phytochrome far red. And so we should keep the equivalent of a long night, so we should get flowering. And so we should get no flowering for the long day plants. Now here we hit it with a burst of red light, which is like a burst of sun, and then a burst of far red light, which means that now we have overridden it and converted phytochrome far red into phytochrome red, and then converted phytochrome red back into phytochrome far red, just like we sustained the night, which means the night goes essentially uninterrupted, even though it was, but it's a double interruption, and so the short day plants should still flower, and the long day plants should not flower. Here, we're going red, far red, red, which is almost the equivalent of here. Interrupt with daylight, convert back to phytochrome far red, convert back to phytochrome red with the red light sunburst, which means we've interrupted the night, so no flowering in the short day plants, and flowering. Oops, nope, sorry, clicked on the wrong button. And then finally, we should go through and we see red, far red, red, far red, that's the equivalent of up here, which means that we should get yes and, oh, hold on. I lost my train of thought, and no. So you are correct. You have determined the effects of these light treatments on the short day and long day plants. A final flash of red light essentially resets the night, creating a large pool of phytochrome far red forms. This form of phytochrome inhibits the flowering in the short day plants, but stimulates flowering in long day plants. And again, let's just go through this one more time. Short day plants, what do you think it is? Flowering or no flowering? Long day plants, flowering or no flowering? Interrupt with red light to convert phytochrome far red into phytochrome red, no flowering. But now we have interrupted the night. Over here, long day plants. That means we are at flowering. Phytochrome far red, or showing far red light onto phytochrome far red will reinforce that it stays phytochrome far red, which means that we are containing the night, which means, yes, we are continuing to have our short day plants flower, and it's just like a long night, so no flowering in the long day plants. Here with the burst of red light, we have converted phytochrome red, or photochrome far red into phytochrome red, but then we burst of far red light, we convert all that phytochrome red back into phytochrome far red, so we have reinitiated the night. And so we will have flowering. So it's essentially the same as having a continuous night, which means no flowering. Here, this is the same as interrupting the night because we have converted that phytochrome far red into phytochrome red, converted it back into phytochrome far red, and then, oh, wait, shoved it back. All that phytochrome far red hit with a burst of red light at the end. And so now all that phytochrome far red is converted back into phytochrome red, which interrupts the night.